PROHITS carry out proteomics at high temperature for single cells. Can you briefly introduce yourself?
Hi all, my name is Dinu Ciobanu. I'm doctoral candidate number 6. I work currently at LSMBO in Strasbourg, France. I was born in Moldova and then I lived for a long part of my life in Italy, where I set my academic background in chemistry. Then before joining the PROHITS project, I had the opportunity to spend a couple of years in a research institute in Italy, building my background in omics-based field.
Could you introduce your project and explain your role within the PROHITS framework?
So within the PROHITS consortium, my main role is to deliver mass spectrometry-based proteomics data. We acquire data on bulk proteomics, so classical shotgun proteomics data, or single-cell proteomics. I'm more focused on a specific project called thermal proteome profiling, where we aim to study the stability of the proteome of the thermophiles across a temperature range, so under a thermal stress condition. Also because they display peculiar features which might be of biotechnological application and overall biological interest.
How does your project connect with the work of other DCs?
So as an experimental scientist within PROHITS, my main role is to deliver data to the whole consortium. Then there's a significant part of computational scientists in PROHITS that do further investigation and exploration of this data. It's also quite interesting and challenging to output specific data like in our cases. For this, we have experienced scientists that are focused on visualizing this kind of data. I am particularly connected with the doctoral candidate 4, Kenneth, that is helping me with outputting this data. Then I'm connected with Josie, doctoral candidate number 2, requesting the samples or investigating and studying the thermophiles that are of interest for the whole consortium. Then I work in the same lab with doctoral candidate number 7, Sunu, and also with the other scientist that is focused on single-cell proteomics, that is Francis, doctoral candidate number 9. But I truly feel connected with all the candidates within the consortium, because gradually all the projects are starting to bridge together. So I also try to connect with other DCs if they have any need of proteomics data.
What secondments or trainings do you have planned, and what are your expectations from them?
So far, we had already 2 different workshops within the consortium where we not only updated about our results, our progress in the research, in the specific research we are focused on, but we also did trainings on bioinformatics, thermophiles biology, which is, of course, of importance for me to have the right perspective on what we are working on and what are the tools that we can use to deeply analyze the data we are outputting. I also had the chance, together with doctoral candidate number 7 and doctoral candidate number 9, to spend 1 month of secondment in 2 different Bruker sites. Bruker is a leader in the analytical instruments field. Especially for us, it's of interest, the mass spectrometry side, of course. We had the opportunity to be trained by Andreas Schmidt and Christoph Crisp, both in Bremen and in Berlin. This was a really interesting moment where we had the opportunity to go into the heart of the factory of the mass spectrometers, which we daily use for our research. Then for other planned secondment, I also will spend an additional month in Debrecen exploring and helping with other proteomics acquisitions.
What motivated you to join this project, and what are you most looking forward to during and after your PhD?
So the biggest motivation for me to join the PROHITS project was, of course, the topic, which was something I was looking for, basically expanding my knowledge in the proteomics field. It is particularly interesting also because apart from classical proteomics, I have the opportunity to be in close collaboration with single-cell proteomics doctoral candidates. This is another big field in proteomics that is currently of hot topic. You need to join a multidisciplinary project where scientists really from any kind of scientific background can contribute and give their perspective on such an interesting topic like thermophiles biology. That's what mainly motivated me to join the PROHITS project.
How do you plan to ensure that your project achieves its expected outcomes?
So for us, both early career researchers and I would say the whole consortium, it is important that we generate enough data and high quality data for meaningful biological insight on something that is quite unexplored so far. So that's the thermophile biology. On my side, as an experimental scientist, I have to provide, as I said, high quality data and make sure that those data are delivered on time, of course. It's also quite important to see the connection with the other candidates, so give insights and information from proteomics data to other scientists. So as an experimental scientist within PROHITS, my main goal is to provide high quality data. This is ensured by using top-end technologies such as the mass spectrometers we have in our lab. We have multiple platforms and we can really envision different experiments, and the one we consider most relevant for a biological insight. And then we can adapt and optimize the methods for a specific question. Also quite important to interpret properly the results that we generate, that we can now generate at high speed and at high rate. I would say that so far we have quite good proteomics results from a selected class of thermophiles. We have at least 5 species on which we can provide proteomics data. It's then up to the whole consortium to decide what's the main direction to take after we realize that we can generate data. Of course, it's of big importance for me to communicate these results and make sure that they are properly delivered and on time for a further exploration and investigation, of course.